I'm Science Officer at the National Botanic Gardens Wales down in Carmarthen. Hopefully some of you will have visited. If not, you definitely should. It's a shameless plug. And yeah, I've been doing my PhD for the last 4 years. I'm in the final intense period of flapping, as someone described it the other day. I'm submitting my thesis next month. So going to talk a little bit about the results from my thesis, but also a bit of the work that some of my colleagues have done. So using DNA to identify the pollen on the bodies of insects, so we can understand more about them. So yeah, as you're all probably aware, insects are really important parts of the ecosystem, but unfortunately many species are in decline. Not all of them, so some of them are doing okay, but the kind of general consensus is that they are overall in decline. And these declines are due to a combination of habitat loss, pesticides, climate change, and increase of pests and disease. And a recent study that came out in 2019 estimated that a third of wild pollinator species have declined in the UK between 1980 and 2013. So that's quite, it's quite worrying, really. So they looked at bees, so wild bees, so not honeybees, and hoverflies for that study, which is what my research focuses on as well. So understanding what bees and all insects really are foraging on is kind of a key component to conservation. If we know what they're foraging on, how this changes throughout different habitats, how it changes over time, then we can really help to provide those resources for them. And something which is often overlooked is the importance of gardens. And so I read a fact a few years ago now when I first started my PhD, and I haven't really stopped thinking about it ever since, which is that the area of gardens, the area which is taken up by gardens in the UK is actually greater than the total area of all of our nature reserves combined, which I just think is crazy to think about, because we put all of this effort, into nature reserves and in conserving them and trying to engage people about their importance, but not many people, do the same things with their gardens. So these are amazing habitats that we can really make a difference in. So that's why I get so angry about plastic lawns and things like that. We need to make sure that our gardens are, as natural and as good environments as we can. So there's a lot of information out there about what plants are good for pollinators, lots of lists. The RHS have a list, the RSPB probably have one, Wildlife Trusts. All of these lists have got a lot of information on them, but they do have some problems. So they're quite inconsistent between each other. A lot of the time they're not really based on empirical evidence. So quite a lot of anecdotal evidence has formed these lists. They don't distinguish between different types of pollinator, and they don't distinguish between different times of the year. So often you get a list of best plants for pollinators and they all flower in July, which is only going to help pollinators in your garden in July. So we're trying to improve the knowledge kind of across the board. But not only that, there's quite a lot of problems with the plants that you can buy as well. So in garden centres, a lot of the plants have been grown in peat, and on the problems with peat, but if you don't know, peat is a massive carbon store, so using it in our compost actually contributes to global warming. And also there has been some research which shows that plants in garden centres have residues of pesticides on them. And we all know the problems to do with pesticides. So we can find out what plants are being visitors by looking at the pollen that they carry. So traditionally this has been done with pollen microscopy, so looking at pollen under the microscope. But this is really time consuming and it requires really specialist skills, which are not are quite hard to come by these days. The pollen is quite harsh, so we can use DNA instead. So obviously the pollen is from the plant that has the plant DNA in it that we can identify. And we identify the plant by using DNA barcoding. So I won't go too much into this, but essentially it's a short region of DNA which is variable enough between species, but not variable enough within species that we can distinguish different species. And we predominantly use markers called RBCL and ITS2, but DNA barcoding is only ever as good as its reference library. So we need to create a reference library of known DNA sequences first. And Wales is really a kind of a good example of good barcoding work, because we actually have a reference library of all of our native flowering plants and conifers, which we sequenced and completed back in 2012. And the nice thing about that it's got really varied habitats, and there was only about another 300 plants which needed to be sequenced, so we could complete the whole of the UK. So that was published this year. We now have this amazing reference library of flowering plants from the UK, which means that you can identify plants from a tiny fragment such as pollen, root, or a bit of leaf. So this is just a quick slide to show the different groups that I'm kind of interested in. So I tell people often that I work with bees and hoverflies. They know the bit about the bees, and then they definitely only tend to remember the bit about honeybees and maybe bumblebees. But we do have a huge diversity of bees and also hoverflies in the UK. So this graph just kind of shows that we've only got one species of honeybee, 24 species of bumblebee, about 250 species of solitary bee, and about 285 species of hoverfly. And we don't really know much about hoverflies and solitary bees in comparison to honeybees and bumblebees. So the National Botanic Garden is an amazing place to study pollinator foraging, because not only do we have the formal garden with over 5,000 plant taxa from around the world, but we're also surrounded by a national nature reserve, so Wynlas, and that makes up a total area of 568 acres. And yeah, so we can really monitor whether pollinators are using these horticultural plants or the more native plants. I put this slide in just as a kind of additional thing, because this is obviously a recording conference, and it was just to highlight really that we've got some pretty cool species recorded at the garden in the last few years. The highlights definitely are Euthera longicornis, so that's a section 7 species, the longhorned bee. So I've got a picture of a female there. I'm not sure if you can see my cursor, but the males have the really, really long antenna, and that was last seen in the county in 2005. And then some other species such as Andrena congruens and Sphacodes scabricolis, which haven't been recorded since the millennium. So I was pretty chuffed with those. So what the kind of basis of my PhD was going out onto transects set up across the garden, walking them every month between March and October, catching bees and hoverflies, washing the pollen off their bodies, extracting the DNA, amplifying the regions of interest, and sequencing them, comparing those to our reference library in order to identify what plants they're using. So I caught 382 bees and hoverflies, and I successfully sequenced 369 samples, which is a pretty good success rate. And just to give a kind of broad overview, I found 191 plant taxa which were used for hoverflies throughout the season. So some of these were identified species, some most to genus, some to family, based on how closely related some of the species are and how difficult it is to use the DNA. But only 6 of these taxa were found on over 50% of individuals, and I of those on the right-hand side. So the taxa which was found most commonly was rubus. We can't identify the rubus to species using DNA because we go for a conservative approach, but it is exceptionally likely that it is Rubus fruticosus, which is blackberry, based on its abundance in the landscape. Then the second most frequent plant was the buttercup and lesser celandine group. So that's just one example of one of the buttercup species. Then we had a group which was composed of thistles, knapweed, and cat's-ear. Now these are quite difficult to tell apart using the DNA, and quite often what we do is when we're manually going through all the sequences, we assess whether things are likely based on when they flower and what habitat and where we know them to be flowering in the garden. And unfortunately, all of these species flower in the same habitat at the same time. So it's quite difficult to tell those apart, but it still gives us quite a lot of information. Then we had Angelica and Heracleum and meadowsweet as well. Now, what I found was that the majority of plants that were used were native and neonative plants. So neonative plants were plants which I'd put to genus level, which had horticulture, also native species. So something like prunus. We've got a lot of horticultural prunuses. We've also got native prunuses. So that was grouped as we just had one category, which was native and neonative. And as the season went on, we could see that the proportion of native plants which were being used decreased as the season went on. And there was a big increase in the horticultural plants being used, which kind of supports the idea that horticultural plants extend the flowering season. So, you know, this time of year in September, a lot of the plants that are still left are kind of your cone flowers, clematis, things like that. And the pollinators obviously use them to replace some of the native plants. I also saw a big chunk of naturalized species here being used, and that is predominantly Himalayan balsam. So that is an invasive species, but a really important late source of nectar, particularly for honeybees. And then when I looked at the differences between groups, I found that there were, you know, quite some striking differences. So don't worry too much about all the different colours on the kind of right-hand side, but what I've got here really is this bar shows the proportion of sequences which were assigned to each plant for Diptera, which in this case was the hoverflies, and then for Hymenoptera. So that's all of the bee species, which were made up of bumblebees, honeybees, and solitary bees. And the key difference here is the fact that the hoverflies use a lot of the Angelica and hogweed in comparison to the bees. So they do use it, but it was less than 5% of the total weeds. The hoverflies use more of the ranunculus and Ficaria, and the bees use more of the thistle group, the Himalayan balsam, which you can see comes from the use by the honeybees, and less of the buttercups. So moving on then to one of my other colleagues' work. So Lucy Richter is just handed in her thesis last month. So she has been looking at commercially available seed mix. So as I said, a lot of the problems that we have with the plant lists are that they're inconsistent and they're not based on a lot of scientific evidence. The same is found mixes. So we really want to know of the species, these kind of seed mixes which are marketed as good for pollinators, what plants are being used in those, and how can we make the seed mixes better. So if you have visited the garden in the last whenever 2018 was, I don't know, everything's a blur since COVID. 2018 and 2019, Lucy had these amazing plots just outside out of our garden where she was trialing these seed mixes, and she had two mixes designed to attract pollinators. So one which was a bumblebee and bird mix, and the other which was a general pollinator mix. And then she had two which were not designed to attract pollinators. So one was an aesthetic mix and one was a native mix. And so the DNA metabarcoding techniques, so to identify the pollen which was used on the bodies of the insects, to see exactly what plants are being used in the mixes. And so these graphs just kind of show the number which were in each seed mix throughout the season. So we've got bumblebees, hoverflies and solitary bees. So throughout the season, each colour relates to the seed mix. So how many bumblebee individuals were on the commercial bumblebee mix in June and then throughout the year. So this is quite a nice example that shows that the commercial bumblebee mix did seem to be the one which was attracting the most individuals throughout the season. And the aesthetic mix didn't do too badly either, but the native one didn't tend to do so well. With the hoverflies, it was actually the native mix which was attracting the most number of individuals throughout the season. With the solitary bees, she didn't tend to find as many solitary bees as bumblebees and hoverflies, so it's quite hard to say which was the best here. And then when she looked at the number of species, with the bumblebees, I'm not sure if I said before, there's only 6 species which are kind of common, so you're unlikely a massive range in the number of species throughout the year. So she didn't find much difference there between the mixes. But with the hoverflies, there are quite a lot of species which are common, and it looked like the commercial pollinator mix was doing, the best throughout the year. And again, with the solitary bees, there wasn't enough data really to make any claims. So then Lucy looked at the proportion of plants which we used in each seed mix throughout the year. So what she did was she did a survey of all the plants which were in flower, and then she compared what was being used compared to what was available. And she found that throughout the season, in all of the mixes, none of the seed mixes had all the plants being used at any point. So the highest proportion of plants which we used was just under 55% of the aesthetic mix was used in July, but as low as probably about 12% of the native mix was used in June. So how can we make this native mix, better in June? And how can we make all of the seed mixes be used more throughout the season? And then Lucy did some analysis which looked at the differences in plants being used by bumblebees and hoverflies. And it was quite nice actually, because she found really similar things to me, even though we had completely different study systems. So what we've got here is the proportion of DNA reads which were attributed to each plant from June right through to October. And with the bumblebees, you can see this massive chunk of purple, which is being used throughout the season, which is the thistle group, so knapweeds. They use the coreopsis and cosmos as well. You've got the bramble here, a little bit of everything otherwise. With the hoverflies, there was no one plant which kind of dominated the season in the same way with the bumblebees. So Lucy didn't have the angelica and the hogweed or the buttercups, but that was, you know, probably due to the fact that these insects really did tend to stay in the plots. But the hoverflies used the Anthemidae tribe, which is a tribe in the daisy family, and it's got things like chamomile and tansetum, which is the English name for that. But these kind of same with the coreopsis and cosmos. So that again kind of supports what we already know to be true, is that the hoverflies do tend to go for these kind of open flowers where the nectar is really accessible, whereas the bumblebees with their long tongues, they can really get into the base of these thistle flowers, which the hoverflies can't. And then finally, Dr. Laura Dawns' work. So Laura's another one of my colleagues who did her partnership with Bangor University. She sampled honey from beekeepers across the UK and then sequenced the pollen which was inside the honey in order to know exactly what plants the honeybees was using throughout the UK. Oh, sorry, I should say, so she sampled from 472 beekeepers and successfully sequenced 441 samples. And was that she found 157 plants in the honey. So most of this honey was taken in July and August, so it's not as long of a scale as some of the other work that we do. But again, just finding the same plants really, and that only 44 of these plants were present in over 5% of the samples. And the top plants were bramble, white clover, brassica, so your oilseed rape, Malii tribe, which is hawthorn, apple, and cotoneaster, sycamore, oak, and then just a lot of kind of a bit of mix of garden plants and some native trees and shrubs and herbs. Now, the really cool thing that we were able to do as part of this work was compare the survey that we did in 2017 to the last national survey of honey, which was done in 1952. And so A.S. Deans, as part of his master beekeeper qualification, I think, did a sampled honey from across the UK and identified, obviously didn't use DNA in 1952, but used this microscopy technique to identify the plants which were in the honey. And what we found was there was a really nice correlation between the 2 years sampled. So honeybees were still using a lot of the same plants that they were using to. So, and the same 4 plants in 2017 with, yeah, the top 4 plants were the same in both years. So that's the white clover, the Malii tribe, bramble, and brassica. So what we did then was compared the abundance in the honey between 1952 and 2017, and we saw some quite stark changes really. So white clover was the most abundant plant in the honey in 1952, and that was significantly decreased in 2017. The use of the Malii tribe decreased. There wasn't any change in the heather, little decrease of red clover, huge increase of bramble in 2017, big increase of brassica, big increase of Himalayan balsam. And that really relates to what we see in the environment as well. So honeybees haven't just, changed their diets in 65 years. They're doing that because the landscape has changed. So what we did then was compared we looked at the countryside survey. In 2007 was the closest year that we could get to 2017, but what we could see was that basically the honeybees were changing their plants in response to what was happening in the environment. So we've had a massive reduction of white clover because less use of clover lays and increased agricultural intensification. We've had a big increase of bramble in the landscape. We've had a big increase of oilseed rape, and 100% increase of Himalayan balsam, which I think was listed invasive in the 80s. So it all makes total sense, really. But it shows how you can use something like DNA metabarcoding and honeybees to really monitor what's happening in the environment. So I've talked a lot about the plants that we put in our gardens and how we can pollinators, but we don't really talk enough about nesting habitats. So it's really important in your garden that you have space for nesting. So a lot of these solitary bees will nest in cavities, in the walls or in bee hotels, or in the ground. So you can make bee hotels, and I've got a blog on our website which is all about bee hotels and different types and how you can make them. And there's a lot of research, particularly on Bug Life's website, about making space for pollinators in your garden by using bee banks and leaving ground, some free ground. And then just to kind of wrap up, I wanted to briefly talk about the Grow in the Future project. So Grow in the Future was it's finishing next year, a 5-year project funded by the EU through the European Agricultural Fund for Rural Development. And that's all been about using gardening to improve health and well-being of people, wildlife and the environment. And through the Grow in Future project, something that's happened to my font there, we have been able to set up the Saving Pollinators Assurance Scheme, which is the first labels kind of scheme of its kind, which uses labels and stickers to let consumers know which plants have been proven to be good for pollinators. So we've gone through all of our research over the last 7-ish years to identify the plants that's coming up in the DNA. We've worked with Wales-based growers, which produce plants in a sustainable way, so not using peat, not using pesticides, and we've been working with 29 nurseries so far. And yeah, it's been amazing to kind of just have a way that your research can really be shown to make a difference, because it's no use doing the research and publishing the papers if only a small portion of the community really engage with it and read it. So I've loved being part of this, and we're really hoping that we can extend it nationally as well. But if you are interested in finding out more about this, then if you go on our website, there's links to the different growers and where you can buy their plants online as well. We've also been selling some seed as part of the Saving Pollinators Assurance Scheme. So we've got amazing hay meadows at the garden, which are really species rich. And we've been collecting seed from them and selling these to organisations, landowners, all sorts of people. And we've had some amazing interest from this. I think we've sold out of all of our seed from this year, but if you are interested in getting some seed, we're only one of 2 providers of Welsh provenance meadow seed. So yeah, definitely email me and I can put you in touch with the right people to start the conversation about that. And yeah, that's kind of the end of my talk. So yeah, diolch yn fawr, thank you.